workshops. And just keep in mind, this session is recorded and will be posted online at a later date. Regarding our agenda for today, I'll be going over some recap material from prior sessions, introducing the background for differential expression, talking about the pseudobulk approach, and then going through the experimental design requirements from Muscat. Then I'll introduce our data set, walk through Muscat, and then talk about FDR options, and then finally just touch on quality control. So in prior workshops, we introduced RNA-seq analysis. RNA-seq allows for the analysis allows you to look at the gene activity under a condition within a cell or tissue at a specific time. And it can be divided into two roads. We have bulk RNA-seq, which shows the average expression that across all cells. And then we have single-cell RNA-seq, which measures the expression within individual cells. So using the fruit smoothie analogy, you take a whole lot of mixed berries and you put them into a blender to make a smoothie for bulk sequencing. And while you can taste that it's a smoothie, you can't distinguish which flavor it came from, which flavor came from which specific fruit. So bulk RNA sequencing allows us to see the overall signal. Whereas for single-cell RNA-seq, instead of blending everything together, you arrange all the constituents separately, where each piece of fruit is distinct. So this analysis allows us to see the source of the signal and is useful in assessing cellular heterogeneity. So here's where today's workshop fits in the bigger picture. We've already gone through this pipeline we talked about planning and pre-processing and eventually data processing in SORAT. And then you'll end up at this downstream statistical analysis step. So today we'll be specifically focusing on differential gene expression analysis. So just going into depth and into the previous data processing step. By this point, your data will have been through quality control, normalization, feature selection, dimension reduction, and especially cell labeling. Cell labels and your sample metadata is what is required as input for Muscat and for differential expression, which is the next step. So what is DE analysis, or differential gene expression analysis? It's a technique that's used to identify genes with significant changes that are meaning have they been up or down regulated. So changes in expression between different experimental conditions. For example, you want to look at which genes are regulated up or down in disease versus healthy samples or stimulated versus controlled samples. You do this by comparing expression levels for each gene across conditions. And you test if the difference is statistically significant, and then you perform a visualization, typically with a volcano plot. You can also further visualize up and down regulated genes as heat maps. So the end result is that you end up with a list of genes with a full change value. That's a quantification of how much it's changed, and a p-value, which shows how confident we are that that change is real. And this tells you which genes and biological processes are driving the differences between your conditions. Now, I'll be going more into this approach. It's known as pseudobulk. Pseudobulk is an aggregation approach to setting up the data before you perform differential expression. It's basically where you combine all of these cells of the same type from the same sample into one expression profile. You're almost recreating a mini bulk sample per cell type per donor. Yeah, so you're deliberately collapsing your single cell data back into something that looks like bulk RNA-seq data, where you have one expression profile per sample per cell type, and then you can use bulk statistical methods. The difference between this and bulk RNA sequencing is that you get one profile for the whole tissue with bulk RNA-seq. With pseudobulk, it gives you a separate bulk profile for each cell type. So going back to that fruit analogy, so you have your individual types of fruit from, like, for example, donor one, you have strawberries, blueberries from donor, and also blueberries. But you blend those together, like individually. So you get a strawberry smoothie, you get a blueberry smoothie, and then you can compare these smoothies between conditions. So now the problem with cell level differentiation or differential expression, on the left, the naive approach of traditional single cell differential expression is that it treats every cell as being independent. But cells from the same donor share genetics in the environment and processing. That correlation means that your effective sample size is just the number of donors, not the number of cells. On the right, with the pseudobulk approach, it solves this by collapsing cells back to the donor level, which is, in most cases, the true unit of replication that we want to look at. So going into how pseudobulk works, you start with an annotated single cell object, and you split the cells by both cluster and sample. So if you have 8 cell types and 16 samples, you end up with 128 groups. You can then sum the raw counts within each group to get one count vector per gene. And then you hand those matrices to your standard bulk differential expression tools like R and RNA-seq. So by aggregating to pseudobulk, we transform a single cell problem into a more well understood bulk differential expression problem. So today we'll be going over a workflow called Muscat. It's a R package available on Bioconductor that provides an end-to-end pipeline for pseudobulk DE analysis in multi-sample and multi-condition single-cell RNA-seq data. It handles preparation, aggregation, testing, and visualization, all in a few function calls. And it wraps proven tools like EdgeR, DC2, and LimaVoom. All of them are built in. So you just pick which one you want to use. And it also contains built-in QC plots, multiple testing correction across clusters, and visualization functions. So this takes this use this package, it requires a SingleCellExperiment object as input, which is basically a Bioconductor data structure for storing single cell data. And this data structure can contain raw counts, metadata, dimensionality reductions, all in the same object. So it's the Bioconductor equivalent of a Seurat object. So as I mentioned, it wraps 3 bulk RNA-seq frameworks, and you choose which one you want to use. By default, you have EdgeR, which works best with small sample sizes. which is usually the most common scenario in single cell analysis. We also have DSeq2, which provides shrunken fold changes, which is nice for ranking genes. Limavoom is the fastest and scales well if you have many samples. In practice, the results are often very similar across methods, and the choice rarely changes the top hits. So before you actually do differential expressions, you need to check your experimental design. Biological replicates means independent donors or independent donors, not technical replicates. So, or not different wells from the same patient. So you need more than two conditions, 3 replicates per group. You need cell type labels and also sample level metadata. Now, just to keep in mind, data sets like the PBMC 3K used in previous workshops, that cannot be used for this type of analysis because that only has 1 sample and 1 condition. So we'll be using the example data set we're using is from Kang et al., which is a pretty well-known example of pseudobulk differential expression. It consists of 8 donors, each profiled under control, and then we're looking at the 2 conditions, which are control versus interferon interferon beta stimulation. It has pretty much everything we need. It has multiple donors. We have clear conditions. We have multiple cell types, and we have well-characterized biology. And there is a way to go from Surat to Muscat, but that I won't be going too much into that process. But there is a way if you want to transform your Surat object into one that Muscat can use. So regarding the Muscat workflow, there are 4 main functions that transform the raw data into results. We have prep SCE, which standardizes your metadata column names. Aggregate data, which sums up the raw counts per cluster per sample. We have PBDS, which runs the actual differential expression test independently for each cell type. And then we have ResDS, which extracts a tidy results table that you can filter and then plot out. And we'll be going more into them in the R markdown. Now, during this workflow, well, you'll also come across FDR, or false discovery rate. This is a statistical method to control the expected proportion of false positives among all rejected null hypotheses. So the expected proportion of your significant results, they're actually false positives. When you test many genes, in this case, some will look significant by chance, and FDR correction adjusts the p-values you get to account for this. So for example, if an FDR of 0.05 means that you accept that up to 5% of your reported genes might be false positives. So there are 2 options within Muscat. You have your local FDR and a global FDR. Local FDR corrects p-values within each cluster separately. So if you test 8 cell types, each one of them gets their own FDR correction. A global FDR corrects across all these clusters simultaneously, which is more conservative. So basically, use the local FDR when you have a specific cell type hypothesis, and it's recommended to use the global FDR when you're exploring across all these cell types you have and want to limit your overall false discovery rate. And then finally, once you get your results, the there's some quality control steps that's recommended. First, make sure that each pseudobulk group has enough cells, as Muscat defaults to filtering groups with fewer than 10. Second, is to make an MDS plot of your pseudobulk profiles to confirm that your samples separate by condition, not by batch. Third, check for library size outliers. And then the fourth is to filter for lowly expressed genes. If your MDS shows batch separation instead of condition separation, then that's when you need to add covariates and perform batch correction. Okay, now we'll be going into the RMarkdown script. Yeah. Stop sharing. Let's see. Yeah. So you can actually find this on our GitHub. Let me just send a link. Let's see. Okay, I posted the GitHub link to the RMarkdown file in the Zoom chat. Feel free to download that. Oh, oops. Yeah, I just sent it. So, yeah, just you can download that, and then just on the GitHub itself. Yeah, if you aren't familiar, you have to go into. You can click download raw file, and it'll just download as an R Markdown, and you can open it in RStudio. Oh, sorry. I can't find. I think. All right. Hopefully you've downloaded the RMarkdown. Um, yeah. So I'll just go through it. Yeah. So let's go through some boilerplate. Yeah. So that's just a setup chunk. Oops. Yeah. So this is where you install all your packages. You need to install. If you don't already have Bioconductor installed, you'll have to do that, and then install Muscat, DSeq2, the SingleCellExperiment, uh, um, scatter, and edgeR packages. And then also, if you don't already have it, you need to install ggplot2, dplyr, pheatmap, and ggrepel. So just run that chunk, and it might take a couple minutes, but hopefully it shouldn't take too long. And then we load the libraries below. Give a couple minutes. It depends on your CPU as well as your internet connection for downloading all these packages. But at the most, it should take 5 minutes. While we're waiting, I can just go ahead to the next steps. Uh, and if it's still downloading for you, just you can still continue to follow along. Um, but it's everything in the script is like self-contained, so should be fine. So the next chunk that we have is where we load our example data. We're using Experiment Hub, which is this bioconductor service that hosts and distributes like data sets. And you can access them or query them using accession IDs. So in this case, we're just uh, loading Experiment Hub and then loading this specific accession ID, which uh, is connected to that Kang et al. Human PBMC uh, single cell data set. And we're storing it in this SCE object. And then if we try to view the if we try to just view the object, this is how it looks. It's a single cell experiment object. That's the class. It's a 35,000 by 29,000 matrix. Contains, these are some of the row names, which are on all different genes. And we have the column names, which are all different, like the actual sequences. Yeah. Now, if you want to do some exploratory analysis and go through the metadata, we can just do a simple head call data, the SE object. And then we can also look at the different using table, we can look at the cell type labels, the sample IDs, the condition, and put it all into one big table where it's we have cells per sample by the cell type. So if we run this, now each of these lines, they give us a different output. We have the data for just the head of the first 5 rows of that of that object. We have the different cell types and their counts. We have the 2 conditions, control versus stimulated, and then we have our different cell types. So step 1 is to prepare the single cell experiment object. So here we're just making a sample ID, unique sample ID per donor and by the condition. I just run this. Yeah, so here's that prep SCE, just to set up the internal metadata that we'll be using downstream. And we're just renaming all of the different columns to cell, sample, and then to the group column, which is healthy versus disease. And then we are also looking at the different or the first 5 rows from that data table. And here we are also doing a log normalization, but to get a log counts. So if we try to do SCE, oh, that's okay. We try to view SCE, and then we're just pulling up that object. We look in the assays, data. Well, there should be a can't find it right now. There should be a column called log counts and log counts. So if we want to do a quick summary of the experimental design, you can run these lines, just looking at the number of cell types, samples, conditions. So yeah, we have 8 cell types, 16 samples in total. We have 2 conditions, and these are the 2 conditions. And then this is just a table showing the cell types and the counts for each. All right, so now we've set up the metadata. You can move on to step 2, which is aggregation. And we use this function, aggregate data. And we're aggregating, as I mentioned before, we have to aggregate through the raw counts instead of log normalized counts, because that is what EdgeR and DSeq2 requires. So we run this. Now we have we've aggregated everything into this PB object, and we've greatly reduced the number of dimensions. So we try to assess this using assay names. Yeah, so these are our cell types. For the first cell type, we have 35,000 genes by 16 samples. If you look at the pseudobulk count for the first cell type, we have these different genes, and then cell types, and these are the different conditions. Okay, now we move on to step 3, which is that quality control step. Here we want to create an MDS plot to check whether these samples separate by condition and not by batch. So this is done through this PB MDS. which is part of Muscat. And here we are, MDS plot. Yeah. So basically, you want to see that all of our clusters here, um, their clusters are distinct from each other. If they were mixed, that would mean that they are um, there are batch effects, and we'd have to include covariates. But because this data set is a sample data set, everything has been there's no need for batch correction. And now we want to look at the cell counts per group. This is just using like a ggplot. We want to visualize the number of cells that contribute to each pseudobulk profile, and also visualize the proportions contributing to each pseudobulk profile. If you run all this, grouping by the cluster ID, sample ID, and the group ID, we get these two. We get for each cell type, we get this large plot showing the number of cells, the X, on the Y, and then each of the sample IDs for each of the cell types that we have. Also separated by the type of condition. And also the same thing for the proportions. So that's just something, quality control step that is useful to look at before you perform your differential expression. NA is just some cell type that they didn't, they couldn't actually like find it, or they just listed all of these samples under this this cell type. I don't think they actually like knew what cell type that belongs to, or like it's just the cell type label. Pretty much, yeah. Now we want to finally run the differential expression. It's done by this function, PBDS. And by default, we're just using the edgeR method. When you run that, just take a minute. And it's done. So you don't have that many samples. It's pretty fast. Now, if you want to extract the results and put it into a tidy results table, use this resds function. And we want to inspect it using head. There we go. Have our different genes, the cluster ID, log2 fold change, average log2 per million, or counts per million, your F statistic that's specific for the edgeR method, raw p-value, and then the adjusted p-value within each cluster, as well as both the local and the global p-adjusted value. Right, and then if you want to look at the number of genes tested per cell, you can also view that for genes per cell type. Now, if you want to filter for your significant genes, because those are the ones that are most differentially expressed, using an FDR value of less than 0.05, log fold change greater than 1. Here is where you can decide on the type of FDR approach FDR correction approach that you want to use, either p-adjust local or p-adjust global. In this case, we're using p-adjust local because we're interested in a specific cell type. We're going to be focusing on B cells today. So if we just filter by p-adjust local, and we just do a summary per cell type, now we have the number of genes that are upregulated, downregulated, and total. These are all different, significantly, they're all significant. And they fall within the thresholds that we had set. Now, final step is to visualize these results. So as mentioned before, for differential expression analysis, typically we want to perform or create a volcano plot. And again, the cell type of interest we're looking at is the B cells. We want to, again, make a data frame for the volcano plot, and then just using ggplot, we're using creating the volcano plot. If I run this, this is how it looks like. Volcano plot between the different conditions. Everything in red is the significant significant genes. Some of them have been labeled that are, or the top hits have been labeled. And this is all under the log 2 fold change. Now, we also can make a heat map of the different top DE genes. We can use this built-in heat map from Muscat called PB heat map, which will just show the top 20 DE genes per cluster. If we just want to run this, there we go. Now we have our heat map showing the control condition versus stimulated, all of the different clusters, that is the cell types, and then the gradient in the different color gradients are the normalized mean log counts for each gene. Yeah, so especially within the state that you're interested in, particularly the stimulated state, these genes in particular, within the cell type, are those are the ones that you may want to look at further downstream. You can also make an expression frequency plot for a specific cluster. You can plot the fraction of cells expressing top genes, and then you can use this function called plot expression, which creates these violin plots. So if we run all that, now we have all of these expression frequency plots for each of your top hits, for each of the conditions. You just look at the log counts for expression. That's something, if you want to look more into this, yeah, it just using this plot expression function. And then finally, if you just want to look at the number of differentially expressed genes across your cell types, like a simple bar chart, you can do that using that same data frame, and you just use ggplot on that to look at the differentially expressed genes per cell type. Just run that quickly. Now we have a bar plot, or yeah, that shows the genes per cell type. We have a lot of genes in this CD14 monocyte cell type. that have been that show upregulation. Same for all of these other cell types. Yeah. Now, as mentioned before, there are alternative DE methods that we can use. By default, Muscat uses edgeR. However, and this is under that PBDS function and step 3, we can change the method type to either using DSeq2 or limavoom if you have a larger, very large sample size and you want to like do it more quickly. So you can do that, and that's the benefit of Muscat, is that it allows for using. You can choose which type of method you want to use depending on your sample size. You can run that, yeah, and it'll save it as another single cell experiment object that you can use for your downstream analysis. I'm not going to go too much into this, mainly because our data set here is very clean. But if you want to add covariates, that you notice from your MDS plot that they're not separating by they're separating by batch instead of condition, you can include covariates. So you need to make sure that the batch info is in the metadata or the column data in your object, using this PB this batch basically column. And then you want to make a custom design matrix based on that using model.matrix, and then make contrasts to for your design matrix. And then once you have that batch attribute, so that's, yeah, once you have that batch attribute, then you can run your differential expression method using the same PBDS, and then you'll get your batch corrected differentially expressed list of genes. I'm not going to run that because our data set doesn't have batches, but in most cases, like raw data, other like with your own data type, that is usually the case. Yeah, I think that is our workshop. If you have any questions, feel free to ask them now. Yeah, that's about it. We'll be again, the RMarkdown is on the GitHub. You can follow along with it. Use it. Go through it on your own later on. Other than that, that's where I'll be ending things. Any questions? Anyone online with questions? Otherwise, that'll be it. So thank you for attending. Hope to see you in the next session of our workshops.